Ovum. It is like any ordinary cell in its structure. It is large oval cell which varies from 117 to 142 microns in diameter. Ovum has two membranes, an inner thin called vitelline membrane and an outer thick transparent membrane named zona pellucida. Corona radiata. It's a 2 or 3 layers of cell which surrounds a zona pellucida when the ovum is shed from. Cytoplasm of ovum is called ooplasm. Nucleus is the germinal vesicle. Nucleolus is called the germinal spot. And transport of ovum in the uterine tube. The ovum passes along by activity of the cilia of the oviduct. Muscular contraction of the tube which increases during ovulation. But the simplest way is to group them according to, and it is like primary membrane. Primary membrane, it is formed within the ovary. Once it is present in the ovary itself, it has a covering or a membrane, and that is known as primary membrane, and it is present between the plasma membrane of the ovum and follicle cells. They are formed either by the egg or follicle cells, as vitelline membrane has been given different names in different animals. Example, in amphibian and birds, it is in close contact with ooplasmic surface until the egg is fertilized, at which time it separates from this surface, forming a tough fertilization membrane. In fishes, it is called chorion. In reptiles and mammals, it is named zona pellucida. So, there's the primary membrane. The primary membrane, it is a vitelline membrane. It is a vitelline membrane. And in mammals or humans, it is known as zona pellucida. Secondary egg membranes is secreted outside the primary egg membrane by a layer of follicle cell that surrounds the oocyte by the ovarian tissue before the egg leaves the ovary. Or it occurs in the form of chitinous shell surrounding the egg in insects and called chorion in the case it have a micropyle for sperm entry. It may secrete female sex hormones as in mammals like membrana granulosa and corona radiata. And tertiary egg membranes formed during passage of egg in the oviduct or during the presence of ovum in the uterus. As the egg of the amphibian, 3 uniform layers of albumin are deposited around it. These jelly envelopes hold the egg together in masses, protect the egg from infection and make them unappetizing to predators. In reptiles, birds, tertiary membranes make up the envelopes of the egg. External to vitelline membrane are added the white of egg, albumin, 2 shell membranes and a porous calcareous shell. And significance of egg membranes: provide the protection to the contents of eggs of developing embryos from different ecological hazards, variable pH, temperature, radiation, pollutions. Prevent polyspermy, that is fertilization by more than one sperm. Once sperm enters into the ovum, the vitelline membrane shows some structural modification, and usually it turns into a fertilization membrane, preventing polyspermy. Help in sperm addition for maintaining normal cleavage of the egg. Unlike the sperm, the ovum is accumulating nutrients and material throughout maturation, not eliminating it. The ovum must provide food reserves until the embryo is able to feed on exogenous materials from the mother. The ovum contributes the maternal complement of chromosomes to the new embryo. The ovum must reject all sperms, but only it should allow entry of one sperm. And the oogenesis of the ovum takes place. It's reduction of chromosome number that takes place by meiosis. The first meiotic division, which is reduction division, that takes place. It's the usually the division starts with mitotic division during fetal condition. Once it attains puberty, by the first meiotic division, each new cell is formed containing a haploid chromosome number. And the second meiotic division usually takes place, completes once the egg is ovulated, or the ova is ovulated from the ovary or discharged from ovary into the oviduct. At that stage, first meiotic division takes place, and then the second meiotic division is arrested at metaphase stage during ovulation time. The first meiotic division that has taken place within the ovary, and the second one that also begins within the ovary, but it is arrested at metaphase stage. And once the ovum is discharged and it happens to fertilize, then only the second meiotic division is completed, once sperm enters into the ovum. So this is how the haploid, the diploid ova is converted to a haploid ovum by the process of meiosis. Oocytogenesis, mitotic division of oogonia, begins during the fetal period. The oogonia enlarge to form primary oocytes. They then enter the first meiotic division, that is stopped in the prophase at the time of birth. The cell nucleus is then transformed into the interphase form, in which it has to stay to the beginning of reproductive life of female. And once it attains puberty, there this first meiotic division continues, and after that immediately the second meiotic division also takes place, but that second meiotic division is also arrested at metaphase stage, and it completes its second meiotic division only after fertilization.